All right, welcome everybody. We're just going to give a couple more minutes for participants to file in, and we'll get started shortly. All right, just a couple more seconds here. Still see some numbers coming in. All right, I think we are good to get started here. Welcome everybody to the 2025 Discovery of Sound in the Sea webinar series. Today we have a webinar on the marine invertebrates and sound. Before I get started and introduce our speaker, Marta, we do have a couple bookkeeping items to go over before the webinar starts. So if you are joining us over Zoom, you should see something like this on your screen. You should see an audio settings tab on the lower left or lower right. That should allow you to turn on closed captionings or transcripts if you would like. And then you should see a chat box button and a Q&A panel button. So throughout the webinar, feel free to ask any questions either to the panelists from the DOSITS team or from our speaker, Marta. If you would like to ask a question, please put it in the Q&A chat box, which should look like this. If you type a question here, someone from the DOSITS panel will answer your question throughout the webinar. And if we don't get to it during the webinar, we will post answers to those questions after the webinar is complete and send those answers out to you afterward. For any other comments or saying hi to anybody else in the chat, please just keep that to the webinar chat so we can keep the Q&A clean for questions. If you are joining us over YouTube, we also want to have your input and questions heard as well. So the DOSITS team will copy those questions into the Q&A, so your questions will be heard and responded to as well. So with that, I'm going to introduce our speaker for today is Dr. Marta Soler from the Laboratory of Applied Bioacoustics at the Universitat Politècnica in Catalunya in Barcelona. She is the lead of the Department of Biology there and researches imaging techniques to assess the impact of anthropogenic noise on invertebrates, fish, and plants. So she is here today to give a webinar on the impacts of marine invertebrates, or the impacts of anthropogenic sound on marine invertebrates. And with that, Marta, please take it away.
Hi, Marta. Sorry, I think you're muted.
Okay. One moment, please. Oops.
We can hear you now. All good.
Can you hear me? Can you?
Yeah, we can.
Yep, we can hear you now.
You can see my screen, no?
Yes, all good.
Okay. Thank you very much for this nice presentation, Christian. I want to thank all the DOSITS teams the opportunity to have this talk today. As he say, I'm Marta Soler, a senior researcher on the Laboratory of Applied Bioacoustics from the Universitat Politècnica de Catalunya. Our laboratory is in Vilanova i la Geltrú, in the south of Barcelona, and my background is as a marine biologist and environmental engineer, and I involved mainly in projects regarding the acoustic impact on marine invertebrates, fishes and plants recently too, through through imaging techniques and proteomic techniques. My talk today is marine invertebrates and sound. Mainly, I want to review all the concepts about marine invertebrates and noise. You can find all the contents that I will say today in a recent review that we write with many colleagues in the field, some 1 year ago, like this. This is called Marine Invertebrates and Noise. Is published within a special issue that we edited that is called Marine Invertebrates and Sound and Frontiers in Marine Science. I know that perhaps the presentation of today has a lot of information, but don't worry, because you will have the presentations, the slides, and you can go to this review to know all the things that I will say. And if you hear me, I will present an overview of the actual knowledge on marine invertebrates bioacoustics. The contents of today are basics of marine invertebrates and bioacoustics, the techniques that you use to study invertebrate bioacoustics, some perception and some production, including receptor systems, marine noise pollution, including sources, the effects of noise on marine invertebrates in different stages of marine invertebrates, populations and ecosystems, at physical, behavioral and physiological level. I will analyze here some of our works, of our papers that we have published in this field to illustrate these different types of effects. And finally, I will try to say some gaps and perspectives. But the role of sound from marine invertebrates, as you know, sound travels 5 times faster in water than in air. This implies a long-distance communication in water, but also implies a long-distance impact of noise on aquatic animals. Sound is an important sensory modality for marine organisms because the other senses, like vision, smell, or taste, are very limited underwater. The scientific knowledge of the biological significance of sound perception and production in marine invertebrates is scarce. They use acoustic signals for communication about predators, prey, territorial defense, social and sexual behavior, and identity. And they detect sounds both as a part of communication and to make use of acoustic cues in the environment. For example, the larvae of corals use them for settlement and habitat choice. Marine invertebrates is a wide diversity taxa that includes crustaceans, cephalopods, gastropods, etc. And this implies a variety of habitats, including the water column close to, on, or within the seafloor. This implies an acoustically complex habitat with multiple routes for sound and vibrations to travel within seafloor and the water column, as well as in the interface between them. There is vibrations in many ways, as you can see here in this diagram of some waves generated by a pile driving. The sound is traveling down the pile, creating waves in the water, the sound waves, in the sediment, the pressure and shear waves, and in the interface between them, interface waves. Waves can transit from sediment to water and vice versa. And in addition, waterborne acoustic energy has 2 components, the sound pressure and the particle motion component. I will come back to this concept later. Let's go to the techniques that we use to study the marine invertebrates bioacoustics. Scientific and diagnostic imaging allows visual representation of invertebrate sensory structures, organs, or tissues for different purposes, like the study of the normal structure, the normal anatomy and function, or the diagnosis of the effects of sound in these structures. Imaging techniques include mainly scanning electron microscopy, that you can see here some images, transmission electron microscopy, like this, and magnetic resonance imaging. Scanning electron microscopy produces images of the surface of the sensory epithelia using a beam of electrons that interacts with the surface and give us information on topography and composition of the sensory epithelia. And it has been used in different taxa to do a description of the surface of the sensory epithelium and the effects of sound on them. In transmission electron microscopy, use a beam of electrons that pass through the ultra-thin specimen and forms an image and is used for the description of the invertebrates ultrastructure of the sensory epithelia, and allowing the inner cellular organelles to be visualized and analyzed the effects of sound on these epithelia. And finally, the magnetic resonance imaging is a non-invasive technique that allows creation of 3D images of body internal organs using powerful magnetic fields and radio waves. This technique has been used to construct models of the morphological structure of invertebrate sensory systems. The next technique is electrophysiology. You can see here an image of an evoked potential hearing test of an American lobster. In this case, these techniques are used in a variety of invertebrate taxa as a measure of the sound sensitivity, and for that we need to put the sensors near the central system, central sensory systems like brain, central ganglia, or near the statocysts. And what we measure are response from neurons associated with sound detection, and has been used for different taxa like squid, prawns, etc. This is the next technique, respirometry, that you can see here represented. In this technique, we assess the effects of the stimulus on the metabolic rate of the animal, and for that we measure the changes on the respiration rate of the animal. Normally the animal is put within a camera like this, and we analyze the changes on the respiration rate by the analysis or the assessment of the level of oxygen on the surrounding water of the animal. Is a measure of the level of stress, and this is an example to use for the adults, and this is an example to use for larvae or eggs, and it has been used for different taxa like bivalves and cephalopods. And finally, we can use the cellular, biochemical, or molecular aspects as a technique to assess the impact on marine invertebrates for sound too. In this case, we have some potential biomarkers of stress like hemocyte count, H2 proteins expression, total protein concentration, phenol oxidase activity, glucose hemolymphatic, and changes in enzyme activities. All of these biomarkers allow us to see changes on the level of stress of different groups of different taxa of marine invertebrates. Let's go to the sound perception, and before that I have to remark some physics aspects. As you know, sound is a physical phenomenon, a mechanical oscillation of particles in an elastic medium capable of provoking an auditory sensation. Sound propagates in the form of pressure waves, like you can see here. The sound waves are variations of pressure which are transmitted through space and time, resulting from the movement of the particles moving themselves from their position of equilibrium. And the frequency is the oscillation per second that are produced in the particles from the medium with respect to their position of equilibrium. Sound pressure can be described by its magnitude and its temporary and frequency characteristics, but in a single point, sound pressure doesn't know information about the directionality of the sound, but particle motion, in addition, have magnitude and temporal and frequency characteristics, but in addition, it always contains directional information because of its inherent movement back and forth back and forth movement. Many aquatic invertebrates use particle motion, including to detect the direction of the source. Underwater heating may involve the detection of the pressure component, the particle motion component, or the detection of either the two sound field components. In invertebrates, there's a lack of information concerning the reception of the sound processes, and the special structures like statocysts and epidermal lines that are able to detect the low frequency sounds. And for that, they have special structures, ciliate sensory cells that act as a mechanoreceptors. Statocysts would work as a low-frequency accelerometer light detector, and in addition would be able to detect the component of the sound pressure too. All of these structures have special features, and this bring me to the biological aspects of the sound perception. The basic structure of the sound perception is the hair cell. Hair cells are mechanoreceptors, sensory cells that detect mechanical forces, and they are specialized cilia. These cilia are the starting point of mechanotransduction processes and are present, for example, in hearing, proprioception, or gravity mechanisms. And they are on the body surface of the animals or could be enclosed in filled cavities like the statocysts. In this scheme that you can see the simplest condition of the hair cell in jellyfish that consists and has evolved in 3 lines, one that goes to Octopus vulgaris, to cephalopods that presents chinocylia arranged in bundles, and the other line that goes to insects presents only one chinocylia, and the third line that goes through chordates through ascidians present only one chinocylia with sterobili. This characteristic is conservative in fishes, in birds, and mammalians, where the chinocylia is disappearing, is progressively disappearing, and they conserve only the sterobilis that are present in the avian basilar papilla and mammalian cochlea. Sometimes these mechanosensory systems present accessory structures like the statolith, the statoconia, the cupula, or the tunic that provoke the fluxions in these sensory cells. Depending on the group, we found different types of hair cells. For example, this is typical from cephalopods. This is from a cuttlefish with the bundles of chinocylia. In the case of snails, jellyfishes, and this is from anemona, they present one solitary chinocylia with the crown of microvilli, and in the case of crustaceans, they have monociliary cells without microvilli, as you can see here. Invertebrates can detect underwater sound through 3 types of sensory systems: the body superficial receptor systems, the internal statocyst receptor systems, and the condotonal organs. As you can see here, all the groups present superficial receptor systems, the lateral line and the flow detectors, and statocysts, but only crustaceans present, in addition, condotonal organs. Condotonal organs are associated with flexible articulations of the appendix. So you can see here, for example, the first walking leg of a crab, and you can see here mark the position of the condotonal organs, but we can find them in for example, in the basal segment of the antennal flagellum of the hermit crab too. The condotonal organs is a proprioceptive organ that monitors joint movement, direction of the movement, and static position, and in some cases could be related with sound perception too. Under my point of view, the most important structure for the sound perception in marine vertebrates is the statocyst, that can be defined as an internal receptor system analogous to the vertebrate inner ear, the otolith organ. Is an equilibrium receptor system and a gravity receptor system, and in some cases, like in cephalopods, it can detect the angular and linear acceleration and the movement of the particles too. As you can see here, it consists mainly in a cavity filled with endolymph, and in the walls there are the hair cells bearing chinocylia, and they have a mass, in this case an statolith, but they are other types. that provoke deflections in these hair cells, and this information goes through the nervus plexus that is around of the statocyst. Statocyst is present in all the groups and present different range of complexity depending on the group. For example, here you can see two cavities of the statocyst of a cuttlefish opened with the statolith, the stone that's in the top, that is in the top of the sensory epithelia, that is below it. In this case, you can see a statocyst of octopus that consists in a bag hanging by strands into the cephalic cartilage with the stone in the top of the sensory epithelia. And this is from a squid, a larvae of a squid, with a small statolith and a small hair cells. In the case of snails, they have a lot of stones, not only one. In this case, we talk about the statoconia, not the statolith. In some cases, the mass can be embedded into the sensory epithelia, like in the case of the jellyfishes, and you can see here the position of the statocyst in the basis of the antenna in crustaceans. Let's go to the morphology of the statocyst. Sound perception I will explain in cephalopods because it's the more complex case. A statocyst, in this case, is located in ventral position into the cephalic cartilage and have two systems. The macula statolith system that you can see here, that consists in una sensory epithelia of hair cells being between 50 and 150 kinothelia morphologically and physiologically polarized in one direction, like you can see here. And in the top of this, there is the statolith that provoke deflections on the hair cells, and in the basis there is the nervus plexus connected with the central nervous system. The other system is the crista cupula that consists in rows of hair cells overlaid by a flexible cupula that has the same function that statolith in the macula. And in the basis there is a nervus plexus too, connected with the central nervous system. When a statolith or cupula provoke deflections in these hair cells, depending on the position, on the polarization of the hair cells, it relays more or less neurotransmitter to the nervus plexus, and it causing an excitation or inhibition response that serves to modulate or regulate a wide range of behaviors. For example, in the case of cephalopods, the locomotion, the posture, the control of the eyes movements, the pattern of the body coloration are responsible from the reception of the low frequency sound waves too. You can see here in this reconstruction of a head of a cuttlefish with the eyes and the arms, the position of the statocyst within the cephalic cartilage in orange, and here are in red the statocyst in the basis of the cephalic cartilage. And finally, the water flow detectors, the superficial receptor systems, are analogous to the lateral line on fishes. They are present in many groups, in all the groups. And you can see here, for example, this is a larvae of a squid with the eye and funnel, and you can see the presence of the lateral line, like a lateral line, it's not exactly the same, in the head and in the arm, and this is a detail of the structure of the hair cells. But you can find them, for example, this is a larvae of cuttlefish with his hair cells in the lateral lines. But you can find, for example, this is a sea lice that is a parasite of the salmons. It's a big problem with many losses in the fish farms in Norway. And this is an adult, and this is another stage, the copepodid, that is responsible for the attachment to the fish. In this case, you can see in the antenna of the adult and in the antenna of the copepodid some hair cells here and all around of the wall. In this case, these epidermal sensory cells carry kinothelia and can be mechanically deflected by local movements that occur relative to the animal's body surface. Let's go to the sound production. Marine invertebrates can produce sound for different variety of behaviors, intentionally during moving or feeding, or deliberately for communication, reproduction, or defense. There is a lot of papers here. You can find all of them with all the characteristics of sound production and the reference where you can look the information in the review. I don't go through all of them. Only, for example, in the case of bivalves, we have mussels that do snapping sounds by stretching its visual teeth, so they have movements. For the valve movements, I will explain later better. The echinoderms, for example, have cracking sounds by stimulation of his spines, but they have a special structure, the Aristotle's lattern, that I will explain later. And in crustaceans, for example, we can find that the snapping ring produce explosive clicks. American lobsters produce caparaz vibrations, or European lobsters produce male buzzing sounds. Let's go to hear some of them. For example, Piurkin, as I say, produce cracking sounds during movements of their spines against a hard structure, but they have a special structure, the Aristotle's lattern, that consists in 5 teeth in the ventral side that they use to scrape algae or invertebrates from the substrate, and they use the calcified caparaz as a resonator. You can hear them. I know there are perhaps some problems of delay, but I will try to reproduce 2 times. You can find all these sounds in the DOSITS web too, these and more. I think you can. They are very nice sounds. Spinolobsters produce pulsatile rasp by stridulation. They use flexible structures to stridulate. They produce a rasping sound using the antenna in a stick and a slit movement, similar when we use a bow moving over the string of a violin. This is called stridulation. When the lobster's antenna moves a piece of tissue at the base of the antenna, the plectrum, rubs against a smooth file located below or near the eye, and it sounds like this. We will do another time. Okay. Another sound is mantis shrimp that produce low frequency rumbles using or vibrating hits caparaz like this. Another meant to do sound is cavitation. It's used for snapping a shrimp. They produce explosive clicks by cavitation when they have a very large claw compared to the other claw, and when they open and close very fast this claw, it's produced a bubble by cavitation, and the sound is produced when this bubble of vapor collapses and produces sounds like this. Another sound is rapping that is produced by ghost crab. Semi-terrestrial crabs can produce sounds by rapping a body part against the substrate. For example, the male ghost crab hit their claw to the ground and produce this sound. And finally, the scallop produce like a sound like a coughing when they expel the water and feces. By the contraction of the two valves, they produce this sound. Is a sharp crack followed by a long puffing noise when the two valves are closed. It's like an explosion, if you can hear them. Okay, let's go to the marine noise pollution. The combination of natural and anthropogenic sounds can create a highly variable soundscape. While marine animals are used to or are adapted to changes in natural and biological sounds, the relative recent introduction of anthropogenic sounds on the sea interfere with essential biological and ecological functions and can cause a wide range of impacts on marine life. You can see in this table, modified from the original OSPAR document, some different examples of sound classified by its sound level, the bandwidth, the measure amplitude, the duration, the directionality, and the sound type. There are mainly two types of sound, the tonal or impulsive and the continuous. Continuous can be defined as a sound with a narrow frequency range that extends over long periods of time and is an impulsive sound is a sound of short duration and a wide frequency bandwidth. An example of impulsive can be piling, air gun, and oxo explosion. An example of continuous could be shipping and drilling, for example, and we can hear some of them. For example, shipping, if we choose ferry, sound like this. It's a continuous sound, very annoying. Another continuous sound is drilling that is produced when we, in the construction of the windmill, is a continuous sound. As an example of impulsive, we have piling, that is another noise that we produce in the construction of windmill farm. And finally, the explosion is another impulsive sound. It's a oxo explosion. These types of noise, this ocean noise, may cause a variety of different impacts on these species. For example, animals may be distracted from finding food, have an increased stress-related chemicals, malformed embryos, or suffer damage. For example, when we use seismic surveys, the number of zooplankton in the sea can decrease, or we can find some strandings of marine invertebrates like you can see here. Let's go more in detail on the marine noise pollution. We will start with the real life stage. As I said before, all of these papers are in the review. You can find them, but some example of the effects could be the light hatching and development, environment, embryonic development, larval normality and mortality rates, the loss of the zooplankton, I don't know, primary settlement of the barnacles can be impaired or have delays in metamorphosis. But in some cases, I say that low frequency sounds could be used for some species to fauna, to orientation, to habitat selection and settlement, especially in corals. And I want to, you can see here our larvae. You can see our cuttlefish, these are hatched and are swimming, but here you can see one moving into the egg. They are the cuttlefish that we use in the experiments. In this sense, I want to show an example of a paper that we did, a work that we did that is called Critical Period of Increased Sensitivity to Acoustic Trauma in 3 species of cephalopod hatchlings. We work with cuttlefish and 2 types of squids. And we analyze the effects on the statocyst and lateral line system. You can see here, for example, the lateral line of a cuttlefish. This is a normal aspect, and the decrease of the number of hair cells with time, but it's known that with increase of time, the level of lesions is worse with time. The novelty in this work is that the severity of the lesions advance more rapidly in hatching than in adult animals. And this picture suggests a critical period of increased sensitivity to acoustic trauma in those species, that has been described before in developing mammalian cochlea and avial basilar papilla. In general, there are 4 categories of ocean noise impacts on marine life: masking, physical, physiological, and behavioral effects. Masking occurs when noise interferes with a sound or signal of interest and decreases the animal's ability to detect, recognize, or understand this sound. And for example, it could be sounds of prey, predators, and mates that are vital to marine animals. In this scheme, you can see that the sounds of some, or the animal hearing range of some marine invertebrates in the top are coinciding with the frequencies and the characteristics of the anthropogenic noise sources, and so that these sounds can mask and in some species, especially in bivalves and crustaceans, provoking the incapacity to use these sounds for communication, reproduction, etc. You have these tables on the paper, on the review for different groups, but mainly the information is that the bivalves have acoustic sensitivity between 10 and 1000 Hz, cephalopods between 400 and 1500 Hz, and crustaceans between 20 and 3000 Hz. Let's go to the physical effects. Intense noise exposure may cause physical injury, including temporary or permanent hearing impairment, and this permanent inability to detect signals at particular frequencies could impair vital life functions to the point that they can go to the death or can impair their survival. The same that as before, all of these papers are in the review. You can see, for example, some effects on the bivalves and the dose-dependent increased scallop mortality, abnormality reflexes. In cephalopods, that is the group where we have been working more, or we have worked a lot in these 20 years. We have been analyzing the lesions on the exposed animals in the sensory epithelium of the statocyst, and these lesions are consistent with a massive acoustic trauma observed in vertebrates, and we have been working in laboratory and offshore conditions. In Nidarians, we found 2 morphological effects, severe damage to statocyst sensory epithelia, and in crustaceans there are very good papers relating the ultrastructural statocyst damages in crustaceans and the impaired writing reflex that this implies effects on statocyst that is related with writing reflex. Writing reflex is the capacity of the animal to recover the upright posture when you put down them. At this point, I will show some examples of our research in this impact acoustic, because our mainly research is in the physics effects. We started working with cephalopods, with these 4 species, and with cuttlefish, 2 types of squids, and octopus animals. We have we collect our animals thank you to the help of the fishermen that bring them to our lab very fast. You can see here the installation on the lab, that is a closed system of recirculating natural seawater. They are fed with natural food, and they put the eggs in the tanks, and we analyze the adaptation to captivity and the state of the sensory statocyst epithelium by the different techniques. You can see here a video that when we feed them, this is happy, this is not as happy because he has not the food. What we did in our first experiments was to expose these animals to between 50 and 400 Hz sinusoidal wave sweeps for a period of 2 hours. And after that, we sacrifice control and exposed animals at sequential times because, as I said, the damage is worse when it's more time after sun exposure. And we analyzed this by scanning and transmission electron microscopy, the statocyst inner sensory epithelium. For example, you can see here the macula statica princeps sensory epithelium in a control. These are perfectly orientated in one direction with all the kinethelia perfectly conditions. When we expose them, what we find is that there is fusion of the kinethelia, there is the apical pole extruded into the into the statocyst cavity. You can see here the loss of, totally loss of the kinethelia or the destruction of the sensory epithelia. Here is totally destruction, all of the hair cells are extruded into the cavity of the statocyst. Here is the formation of some scars, thank you to the supporting cells. And when we go to the transmission electron microscopy, you can see here a normal epithelium with perfect structure. This is a hair cell with all the inner structures, the ultrastructural organules perfectly, like the nuclei, the endoplasmatic reticulum, the Golgi, etc., and the structure of the kinethelia perfectly. And when they are exposed, you can see the totally the structuration of the sensory epithelia and the nervous plexus in the basis too, the expulsion of the apical pole of the hair cell and the closing of the supporting cells forming an scar, and the expulsion of the inner material forming white zonas or dark areas. This is the first time that we show that the sounds provokes physical lesions on the sensory epithelia of the statocyst. After that, we did some experiments at sea, and for that we put the animals at different depths in cage, and the controls were 2.5 kilometers of distance, and we exposed at the same sounds that in laboratory conditions, and we control the particle motion and the level of pressure, and after this exposure we found exactly the same results. These are the control animals, and you can see the same lesions, the fusion of the hair cells, the extrusion of the inner material, the expulsion, total expulsion of the hair cells, or the expulsion of the apical part of the hair cell. And for that, these experiments offshore validate our previous results in lab conditions and allow us to continue working in lab conditions. After that, we work with some applied studies, applied researches. For example, we are asked to see the impact, the noise influence on cuttlefish in the construction of a windmill farm in Saint-Brieuc in France, in the English Channel. And for that, you can see here, I want to point here that it's very important when we assess the impact of sound to use different techniques for assessment, because perhaps you don't find any effect in behavior, but when you go to the physical or physiological, you can find effects. This is the case. In the case here, you can see the cuttlefish below of the transducer, and when we put the continuous sound, they have no reaction, and they are just below of the transducer. But we have, when we went to the analysis of the sensory epithelia, we found lesions on the lateral line on the statocyst in adults and larvae, and in pressure level and due to pressure and to particle motion. And we found effects on the level of eclosion and the level of hatching of the eggs, and in the level of survival of the larvae. And obviously these effects were big, very big, near to the construction, and they were disappeared when you go far from the construction. Obviously, it's for that I say that it's necessary to do a wide range of assessments. After that, we work with other species like jellyfishes. This is the status of jellyfishes. We found similar results. These are the controls, and you can see here the fusion of the crown of microvilli and the loss of the kinocilia, or totally loss of the hair cell. We work with hermit crab and with the attached anemonas in Hiscaparas, and we found similar results. These are controls in the 4 groups of hair cells into the statocysts of hermit crab. And when exposed them, we found loss of hair cells in Zeta, that are the specific hair cells of the crustaceans. You can see loss of the hair cells, or totally loss of the Zeta. And when we see the anemona attached in the caparas of the hermit crab, these are the controls. And you can see here the kinocilia with the crown of microvilli, and we can see crown of stericilia loss or totally fused, then a flaccid kinocilia, or hair cell partially or totally ejected from the sensory epithelium. After that, we applied this research to fight against pest. In fact, apple snail is a pest for us because it arrives here with boats, but is originally from America. It's not from the Mediterranean area, and he arrives here, and he's attacking all the rice fields in Delta Ebro near the Barcelona. In this case, we try to attack this pest. You can see here with sound, you can see here the normal epithelium of control, and after apply the sounds, you can see similar results that in cuttlefish and in the other species. The loss of kinocilia, the extrusion of total extrusion of the hair cells, some hairs in the bases, etc. Another pest that I had said before is the sea lice. You can see here a salmon with the adults attached to the head. And as I said, they have a cycle, this cycle. You can see here the copepodid that is responsible from the attachment to the salmon. And what we want to do is to impart damage on the antenna that are the responsibles of the perception of the sound, because they don't have statocysts. They are floating on the water, and they don't need the statocysts to perceive the gravity perception. And after expose them on laboratory and in field conditions, in the case, what we found is that with time, the hairs of the antennas are fused, and with time they increase this fusion until totally fusion at 72 hours after some exposure. For that, we think that is a good method to fight against pests. We have 2 patents to fight against this pest and another pest, and we think that this is a good way to fight. Obviously, we see if there are some effects on the fishes, on the Norwegian salmon, and for that we analyze the otolith organ by scanning electron microscopy. We didn't find any lesions on sacula, tricula, and lagena, and the 3 sensory epithelia of the fishes. No effects on lateral line, and no effects on the histological assessment of the different tissues of the salmons. And for that, I think it validates our method like a good method to fight against pests. Let's go to the behavioral effects. As you see, there is a lot of papers on that. You can see this in the review. I think this is the technique that has been used more to see the effects, but I remember that we have to use different techniques to not lose information. Behavioral effects can include changes in acoustic communication or vocalization, problems in feeding or social interaction, changes in movement or living behavior, and temporary or permanent habitat abandonment. I want to show you one experiment, one work that we did with blue crab. Blue crab is a best for us too, because it's originally from America and arrives here with boats too. And what we want to do in this work is to see if there are some effects, so some cross-sensory interference. If you between some cross-sensory interference after exposure of the animals to noise, they have they smell the food, and the olfactory sensory system is in the antenna. They rub the antennas to find the food, and in the basis of this antenna there is the statocist that is responsible from the sound perception that you can see here. They are very connected, and in addition they are connected to the eye that is very near us. That with this experiment we want to see if it's affecting one of these systems, for example the statocist, the other systems are affected. For that, we expose the animals in a big tank that we have in the laboratory, and after that, we put the animals into a labyrinth to see if the capacity of the animal to find food is affected. And as you can see here, the animal is here, the crab is here, all the 4 ways are closed because he needs sometimes for habituation. He's rubbing his antennas to detect where is the food. We have one dispenser of food in every score, but here only these have the food. And this is another example that we have to do assessment of different things, because in this experiment we didn't found now it's moving. We didn't found statistical or significant effects on the capacity to find the food, but we found effects on the capacity of on the writing reflex, on the capacity to write. As you can see here, he found the food and he ate this. But there is not significant effects on the capacity to find food, but yes, in the writing reflex, and this implies effects on the physical effects on the statocist. Let's go to the physiological effects. There is a lot of papers, good papers, and the effects could be increased stress, changed metabolic rates, weakened immune system, reduced reproductive rates, and long-term health impacts on marine animals too. You can see there are very good papers in the valves about decreased respiration rate, alterations in metabolic rate, in crustaceans, alterations in respiration, in equiderms, significant changes in enzyme activity and in gene and protein expression. And we have a paper on cephalopods where we analyze the changes in statocys endolymph, in the protein content of statocys endolymph, as a biomarker of stress. For that, we want to assess the structural effects of low-frequency noise exposure on statocys endolymph proteins that act as a biomarker on adult individuals of cuttlefish using proteomic techniques. For that, we extract with microsharings the endolymph of the 2 statocys of cuttlefish at different times, and we analyze them using 2-dimensional gel electrophoresis coupled with mass spectrophotometry analysis, and we identify 900 proteins within the statocys endolymph. And by comparison of control and exposed animals, we found differential expression on the affected by sound animals. Mainly the proteins are related with a stress reaction, with the oxygen transport, or with antioxidant enzymes related with redox state regulation processes and cell signaling, or related with a structural function like cytoskeletal proteins. This is normal because there is a big destructuration of the sensory epithelia. Or another reaction like related with metabolic processes like glucolysis, proteolysis, and protein synthesis. This is an example of how biomarkers can be used to see the impacts on the physiology impacts after noise exposure. I want to show you now a now device that we have developed on the lab under Saturn project, in an European project that allows us to identify and quantify physiological, pathological, and behavioral responses of different species of marine invertebrates associated to sound. Assessing particle motion and acoustic pressure separately. This is the device that you can see here, and mainly it consists on a system made of a pair of shakers that enclose at both ends a transparent chamber equipped with different sensors to measure sound pressure, particle acceleration, water temperature, and oxygen content. The input of the energy that the animal that is into the chamber can be configured in two ways, into configurations, the particle motion or acoustic pressure way configuration. So the aquatic organisms receive a higher dose of kinetic energy compared to potential energy or vice versa. In addition, the transparent acoustic chamber allows, if there are some changes on the behavior, and the respiration module that you can see here allows us to see the changes on the respiration and allows to see if there are some changes on the stress level. This is a unique setup that provides us with a multimodal approach for high intensity, low frequency vibroacoustic exposures. This is one example of the results that we can obtain with this device. This is the percentage of unhatched eggs at 3 different exposure levels in PM mode. There is no difference between the lowest level, but there is difference in the higher dose levels. The pressure shows a consistent response versus those, but but the velocity is not as consistent, and this could indicate a stronger effect related to pressure than the velocity. It is possible to fit a dose response curve of the PM acoustic pressure data that you can see here. And finally, there are the effects on population and ecosystems. You have all this paper on the review, but mainly they can have effects on the impact on regional population structure because of the induced emigration and balanced prey-predator relation, effects on larval development that led to a reduced recruitment, different avoidance behaviors like the starting response on populations that migrate from the areas where seismic surveys are conducted, for example. I want to show a work that we did, is very recent, with Posidonia oceanica. Posidonia oceanica is the only superior plant that we have in the Mediterranean. It's in the basis of the ecosystems, and in plants, sound is a channel for short-range signaling involved in modulating root growth. They have columella cells that you can see here in the root cap that are used for gravitoprists. These columella cells have amyloplasts that are plastic, are cytoplasmatic organelles that produce and store star grains that are used for gravity sensing. The sedimentation of these dense amyloplasts with their inner star statoliths triggers gravity signal transduction that promotes the differential cell growth, allowing the root to direct itself downward. In addition, the green plants have in star grains in other parts, like in rhizoma, in the case of Posidonia, in rhizoma horizontal and vertical. And in this case, they use them like a reserve of energy, is for storage, for energy storage, and for the process of nutrition. The amyloplast role in the orienting plan in the water column is analogous to the marine invertebrate sound sensitivity statosis, and probably have a role in sound and vibration reception. When we exposed the plants, we found that the number of star granules progressively decreased after sound exposure and their total disappearance, and we found strange structures when we analyzed them by TEM, by transmission electron microscopy. In rhizoma, the same. The number of ester granules progressively decrease after exposure until their total disappearance, and the remaining ester grains present holes or strange structures. And in addition, plants, especially not all plants, but Posidonia oceanica has in a symbiotic way fungal fungus in the roots that they help Posidonia to do the nutrition, and we exposed the plants, and in addition we found some effects on these fungal leafas that are within the roots of the Posidonia. Its cytoplasm was progressively alterated until totally empty cytoplasma at 120 hours after sun exposure. And for that, I think that the impact on this very low level in the food chain can impact all the food chain and all the habitat and all the community and the ecosystem. And finally, let's go to the gaps and perspectives. The biological mechanisms of sound detection and production lack of descriptive data for most species. Some marine invertebrates groups are very poorly investigated, especially that are especially vulnerable to noise. The physical and physiological variables related to stress, energy metabolism, and hormone responses need to be improved, including proteomic and metabolomic methods. More information is needed to predict the noise responses of understood species or that survive in noisy habitats or have lower auditory sensitivity. There is a need to undertake and compare large-scale and long-term fields and laboratory studies. Adaptation and habituation to long-term noise exposure or a potential recovery from chronic noise exposure has not been investigated. We need a standardized protocol in future publications that should always include duration, frequency range, weighting, filters applied, reference pressure used, source and received levels, distance and duration of recordings, including data on the magnitude and direction of particle motion respect to the source. When performing field studies, a previous characterization of the local soundscape should be provided. The interaction between different stressors, in addition to noise introduction, we have to consider artificial light, habitat, fragmentation, global warming, acidification, etc., and consider the interaction between them. And finally, the response data is necessary to provide regulators and decision makers with proper information. And that's all. Thank you very much for your attention, and I hope you have enjoyed.
All right, thank you very much, Marta. That was amazing. We do have a couple questions in the chat for you. Well, we have time for a couple of them before we have to wrap up today. So we have a couple questions about the sounds that you showed, that you presented on the sounds that invertebrates make. And some people were wondering if these sounds were explicitly related to communication between, say, two different squid, or is it related to some other process?
I doesn't remember, but for example, I think the semi-terrestrial crabs use to communication. I think to find the females is for reproduction, for example. The other one, I don't know exactly as rim, but I think is for communication too. And spiny lobsters are for communication too, to find the female. And sea urchins, I think, I'm not sure if it's voluntary, because I think it's a result from the scrap, the algae invertebrates when they feed. I doesn't remember is this anymore. The mantis shrimp, yes, I think it's producing by the males. I think it's for reproduction too. But I'm not sure because it's not my specialty, but you can find this information in the page of DOSITS that I think is very well explained.
Thank you for that answer. We have time for one more, and I'll try to summarize. There are a couple of questions asking about the sensory tissue damage that you presented as well. And some are wondering if the tissue damage is something that the animal can heal or recover from over time, or is this a permanent loss of sensory ability?
In the sensory epithelium. Okay. Well, there is some works that show recovery of these lesions, previous of our works. I think in fishes. In fishes, there is some works relating this, but I never found recovery on the sensory epithelia of the statocysts. On jellyfish, on cephalopods, on all the species that I have been working, I never see it. I'm not sure because I doesn't work a lot with crustaceans, but I think there is some work that show some recovery, but I think there are others that doesn't see recovery. I have not constance that in marine invertebrates have recovery when they are damaged by sun exposure.
Very interesting. It's interesting. I assume that's a really difficult thing to study. Unfortunately, that's all the time we have for questions. We need to start wrapping this up. So thank you so much, Marta, for your presentation today.
Okay.
If you don't mind stopping your screen share.
Okay. A ver, sincero.
You are still sharing your screen.
I'm sharing. I'm not sure I can. Okay. No puedo.
It should be by the window where you see like the Zoom participants.
I'm sorry.
No, it's okay.
Now?
No, there should be a button to stop sharing. Oh,
Okay.
there you go. All right, perfect. Thank you.
Okay.
No worries. All right. So just to wrap up here, thank you to all of our participants for today. And thank you to Marta for your presentation. That was very interesting. And we got a lot of really interesting comments in the Q&A. So if you did post a question in the Q&A and it was not answered during the chat, we will be collecting all of those questions and having Marta answer each one, and then we will post the answers to these questions on the DOSITS webpage along with the recording of today's webinar. With that, thank you to everyone who participated in today's webinar. If you registered for today's meeting using the link on the DOSITS website, you will receive an email with details about the professional development certificate that you get if you attend all 4 webinars in the 2025 series. And then as well, please take the webinar survey that will be in that email as well. It just helps us keep improving things on these webinars and making them better for all of you. And then also finally, there is a questionnaire that was linked to in the chat. I believe that link will also be either in the email that you'll receive or will be posted on the DOSITS webinar page afterward, after we upload everything following this webinar. So just keep an eye out for those. And if you are planning to attend all 4 2025 webinars and you are planning to get that professional development certificate, please join us for our next session, which is on June 4th at noon, so the same time. And that webinar will be on wildlife and offshore wind and the effects of noise on wildlife and offshore wind. So looking forward to seeing as many of you there as you came today. So with that, we'll conclude today's meeting and enjoy the rest of your day.
Thank you. I stopped the recording.
Okay, so I'll stop sharing.
Thank you, Marta. That was fantastic.
I'm sorry, I think I talk a lot, but I want to summarize all the concepts. I think it's a good way but
It was so good. And we had so many people attending, so many great questions. We will follow up with the questions that we didn't get to answer live in a Word document. And if you wouldn't mind helping respond to those questions, then when we
Yes.
get that finished, we'll post that to the website for everybody to see if they go back and watch the recording later.
Okay, thank you. Thank you very much for the opportunity to have this talk.
Oh,
Yeah.
it was fantastic.
Yes, thank you so much. A lot of really good questions in the chat as well.
Yes, okay.
People were really
People were into it. And we had people on YouTube watching live, and we had people here. We had
Okay, fantastic.
people with you today. It was great.
Thank you very much. Okay.
Thank
Thank
you.
Thanks,
See?
Marta.
Invertebrates, we're out there. We love them.
We did it. See you on Aquatic Noise.
Yes, yes. See you in
Yes.
Prague.
Thank you.